Hello AP Biology students, we're going to go over the third section in Unit 6. And in this section we're going to cover something called the replication of DNA. You might remember talking about replication when we talked about mitosis and how the cell preps itself to divide. That duplication or doubling of DNA is really important for new cells to have an exact copy of the original DNA. That whole process is going to be covered in this section. Now back to the history of DNA. When we were first trying to figure out what the replication model of DNA was, we had a couple different hypotheses. As you can see, we had something called the conservative, the semi-conservative, and dispersive model. In all three of these models, the old strand of DNA would be a template for the new strands of DNA in some way. Now, I'm not going to get into all of the details of the experiment, but basically we figured out that the DNA replicates in a semi-conservative model. That means that each old strand will be used as a template for the new strand. I absolutely love this diagram of DNA that's showing how DNA replicates through a semi-conservative nature. You can see in our original strands of DNA, the DNA is going to be yellow, and you can see that each new strand then is going to be red. That's because each side of the DNA double helix is going to be used as a template for the new strand of DNA. You can see in the second replication, the new strands of DNA are green, and in the third round of replication, the new strands are black. So again, all we're talking about when we talk about DNA replication is just the process of producing two identical replicas from one original strand. And if you remember, this occurs during S phase of interphase. Now, basically what we're going to be taking a look at in this section is this process of replication, because there are a lot of different proteins and enzymes involved in replicating the DNA. One of the first enzymes I like to cover in this section is called topoisomerase. This is an enzyme that basically helps unwind and relaxes the twist in the DNA. The second enzyme involved in replication is called helicase, and this is the enzyme that's going to break the parent strand of DNA apart. Specifically, it's going to break the DNA hydrogen bonds between each nucleotide. You can see in our diagram here is helicase unzipping the DNA, allowing for each strand kind of go into a separate direction. Now we have to know about something called the origin of replication and something called the replication fork. The origin of replication is the location where replication begins. So you can see here in our diagram, this is the origin of replication. I like to think of this as the starting word in a book. It's where everyone begins to read the book at. This is the starting point of replication. The replication fork is going to move as the helicase moves down and unzips the DNA. In our diagram here, we actually have two replication forks. There's one here and there is one here. The replication fork is like where you're reading the book. Say if you're in chapter 4, that's where the replication fork is. It's where the actual helicase is breaking the bonds at. Our next important aspect of replication is something called single-stranded binding proteins. These are sometimes known as SSBPs, and what they do is they help keep the single strands of DNA from getting damaged, cut, or tangled with each other. You can see in our diagram here, these single-stranded binding proteins are on the backs of the template strands of DNA. Our next enzyme that's really important to remember is called primase. Primase is an enzyme that's going to place an RNA primer for DNA polymerase to bind with. This RNA primer is almost going to act like a flag for the DNA polymerase to signal it for to bond to a certain location on the template strand of DNA. You can see here this primase enzyme adding the RNA primer here to the DNA. Now, the RNA won't end up in the final strand of DNA. It's not like DNA is also composed of RNA. This RNA will later be removed. However, this RNA is initially used for the DNA polymerase to bind to the DNA with. Now, for my classes, I'm going to dive a little bit deeper into the DNA polymerases and identify the couple different types. This one here is called DNA polymerase 3. This is the enzyme that pairs free-floating nucleotides to their complementary base pairs. That is A with T and G with C. As you can see, DNA polymerase is binding these free nucleotides to the complementary strand of DNA. DNA polymerase makes in a 5 to 3' direction. This is really important to remember. It's making in that direction. It runs along the 3 to 5' direction. So as you can see here, we have our DNA polymerase. It is making in a 5 to 3' direction. It runs along, though, the 3 to 5' direction. Since this only runs in one direction, we're going to create two different strands during replication, the leading and lagging strand. The leading strand is continuously made. The DNA polymerase moves towards the replication fork. This is going to be in contrast with the lagging strand that's made discontinuously. It's made away from the replication fork. The lagging strand is going to make these smaller sections known as Okazaki fragments, since as the replication fork opens up more and more of this DNA template, more is going to be available to be replicated, so we're going to create these Okazaki fragments. Another type of DNA polymerase that's going to be used in this replication process is called DNA polymerase 1, in which this enzyme removes the RNA primer and adds DNA nucleotides to those locations. So like I said before, RNA isn't incorporated into those DNA strands, it's removed by DNA polymerase 1. One of the last enzymes we're going to learn about is called ligase. This is just an enzyme that binds the new strands of DNA together. You can see there's almost like a kink or a missing bond here. You can see DNA ligase is going to fix that and bond these two nucleotides together. One last thing to go over about replication and DNA polymerase 3, it's very accurate. It only makes one error between 100,000 and 1 million base pairs. And this does sound like it's really accurate, which it is, but if you think about the billions of base pairs that are in our cells alone, that would mean there's a lot of errors that could arise just from one round of replication. So what DNA polymerase 2 does is actually will proofread the DNA and check for errors. This will lower the error rate of replication even further. There's also something called nucleotide excision repair, where an enzyme will come in when it finds an error, take out that DNA, and replace it with new DNA by allowing DNA polymerase to fill in the correct nucleotides. When all this is said and done, this reduces the error rate to about 1 in 100 million to 1 in a billion. And we're going to end with this great meme, DNA after replication. Yep, you get 2 exact copies of DNA.